It's my special pleasure to introduce Don Helinski for our Profiles in Discovery lecture. And I thought I'd say a few words about his background, about him. And I know he's not preparing to start speaking until about 30 minutes after the hour. Don received his PhD at the Western Reserve Medical School in 1960. And he then joined Charles Janowski at Stanford for his postdoctoral work, which addressed and answered one of the central questions of modern genetics, exactly how are proteins encoded in DNA. The answers contained in a series of publications leading up to the 1964 paper in PNAS, of which Don is a co-author, entitled On the Collinearity of Gene Structure and Protein Structure. Sidney Brenner and his laboratory at Cambridge approached the same question by a different route and reached the same conclusion in a paper with almost the same title, published at almost the same time. But by the time of that publication, Don had moved to Princeton, his first faculty position, and he stayed there in the Department of Biology until 1965, when he came to our new biology department, very soon after the completion of the first buildings, the Revelle College buildings on the upper campus. At that time, there were, by different count, 6 or 8 members of the faculty. So there's been a growth spurt since that time. That's probably matched by the number of real estate agents in La Jolla, but both are really quite remarkable. Don's 41 years here are a proud part of the history of biology at UCSD. It's here that he conceived and developed his explorations of the world of plasmids and their circular DNA, his inventions relating to cloning and the manipulation of gene content of plasmids, and his analysis of plasmid replication and how plasmids are maintained in their bacterial cells. It's work that I think delights and surprises for its incisiveness, its elegance. It's work that also has led to one of the iconic images of current biology and genetic engineering, the luminous plant. But I think we may hear something about that from Don if he has the opportunity. His discoveries and contributions have been recognized with election to the National Academy of Sciences, the American Academy of Arts and Sciences. He's a fellow of the American Academy of Microbiology and an elected foreign associate of EMBO, the European Molecular Biology Organization. At UCSD, he's been the teacher of our undergraduates and graduate students, the thesis advisor of more than 25 PhD students, of dozens of postdoctoral scholars from all over the world, and friend to colleagues in biology like myself. He was chair from 1979 to 1981, and it was during that time, in a particularly difficult and troubling episode involving scientific misconduct, that I got to know Don not just as a brilliant scientist, but as a person in whose integrity and sense of responsibility and devotion to the ideals of the university, I was able to place my complete trust, an unquestioning trust. And I think that that's the way he's seen by everybody that I know. It's also appropriate for Don to give this a lecture in this room because this is the house that he built. He was instrumental in the construction of the Center for Molecular Genetics. He was its director for the first 10 years or so. And I want to add that the years that we shared on the third floor of this building have been my best years at UCSD. His lecture is on plasmid and gene cloning, present at the beginning. Don, you have a chance now.
I could have had as a subtitle to my talk, present in the early years of molecular genetics, and my postdoctoral work was with Charlie Helinski, an extraordinary scientist. I was involved in a project that was trying to answer the question, is there a collinear relationship between gene structure and protein structure? With me, there are two other postdocs, Bruce Carlton and Ulf Henning, and later John Gist. Charlie had produced a large number of mutants of the A protein of tryptophan synthetase and mapped the mutational site of these mutants, working with Jeannie Horn, assistant, and using the P1 transduction system. It was our job to identify the location of the amino acid substitution in the mutant proteins and the nature of those substitutions. And indeed, there was a wonderful correlation between the mutational site map and the position of the amino acid changes. Much else came from these studies, including studies on gene suppression of mutations. And in fact, Stu Brody, a member of our faculty, was a graduate student at the time in Charlie's laboratory. Now, Charlie, we would have our one-on-one weekly meetings, but in addition, he asked each of us to present work in the general area of microbial genetics, molecular genetics, but outside the immediate research field of our interest. So we met on Monday evenings, and after the presentation, we'd be invited to Charlie and his wife Carol's house for a spirited game of croquet, which I think he arranged so to sharpen our competitive skills. In any event, it was my turn, and for whatever reason, I came across some papers on cholecinogenic factors, or plasmids, and colicins. Now, toward the end of last week, I get this call from Stu Brody. And Stuart says, I've been looking through some old papers, and I came across an outline of your talk for that Monday evening, which was in January of 1962. I totally forgotten that I prepared such an outline. In any event, what I talked about mostly that Monday evening were some papers by the sort of father of cholecinogenic factors and colicins, Pierre Frédéric. You can't read that. You have to apologize for a number of my slides. We're going back to some very old papers and some very old data. Pierre Frédéric is a Belgian scientist who did a lot of work on a number of different cholecinogenic plasmids. So I should give you some idea what we're talking about when we're talking about cholecinogenic plasmid. I should also say that back in the days when I was a graduate student, you were required to show proficiency in scientific German and scientific French. And like the other graduate students, I thought this was a total waste of time, and I think for most purposes it was. But it did help me in digging out these papers published in the Institute of Pasteur, and most of the papers were in French. Frédéric, of course, is a French-Belgian. And so it did help without depending upon a lot of other people to translate for me. Now, what I have here is a culture dish with bacteria that allow the development of colony, and these bacteria have a cholecinogenic plasmid. And one of the characteristics of cholecinogenic plasmids, which is why they have that name, is that these plasmids code for and excrete a antibiotically active protein. So after allowing these colonies to develop, one takes a soft agar, which is seeded with a bacterium that is sensitive to the colicin, allow that to develop, and where you do have a cholicinogenic plasmid producing a colicin, it's excreted into the medium, it kills the overlay of a sensitive bacterium, so you get these clear regions, what we call lacunae. And those were the markers that we worked with this system. Early in 62, I did get an offer to join the faculty at Princeton University as an assistant professor. And of course, began to think about what shall I do? I certainly didn't want to work with tryptophan synthetase and compete with such a formidable figure as Charles Janowski. So I wrote up a grant application to study these cholicinogenic factors and colicins, and I wrote to Pierre Frédéric requesting a number of cholicinogenic strains. My grant application was funded for a 3-year period, and in today's dollar, I guess this would be something like $200,000 a year. Now, what makes this remarkable by today's standards is that I had absolutely no experimental experience with cholicinogenic factors and colicins. No person in his or her right mind would write such an application today and expect to get funded. But you must remember, this was in 1962, and this was post-Sputnik. In 1957, the Soviet Union put into orbit this grape-like structure, which I think I read was 183 pounds, and it orbited the Earth 98 times before it plunged into the atmosphere. Well, this threw great fear into many in the American public and certainly at the government levels. And it was decided that we were woefully way behind scientifically the Soviet Union. We must plunge lots of money into the natural sciences, physics, and biological sciences benefited from this. And that helped me a lot in terms of getting, I was always grateful to the Soviet Union for putting that Sputnik up. So, in an event, I went on to Princeton and had 2 wonderful postdoctoral fellows, Phyllis Kahn and Fred Hickson, and a graduate student, Jeff DeWitt. And we set out to purify the hallucinogenic plasmid that I selected to work on. And this hallucinogenic plasmid is chole-1. And we worked with two plasmids here, collagen factor V and chole-1. Collagen factor V is a sex factor. It promotes conjugation between bacteria, and it's able to transfer the non-sex plasmids like chole-1 to other bacteria. At this time, Matt Messelson and Paul Doty's laboratory established that the DNA of a given species or organism was unimodal in its GC content within a narrow range. And the reasoning went as follows. If we could transfer this chole-1 plasmid from E. coli, which had a 50% GC content and a buoyant density, the DNA is 1.710, to P. mirabilis, which is 38% GC and a buoyant density of 1.700, and a cesium chloride gradient, then we could distinguish between the plasmid and the chromosomal DNA. So Bill DeWitt and I published a paper saying that indeed the hallucinogenic factor E1 was of a density of 1.710, like the chromosome of E. coli. It's a natural, E. coli is its natural host. And using equilibrium centrifugation, we were able to pick up a blip of chole-1 at 1.710, and the chromosomal DNA of P. mirabilis is 1.704. At this time, this is, I think, just the third publication from our laboratory at Princeton, I received the call from John Singer. John had taken over the chairmanship of our biology department then, after the untimely and tragic death of David Bonner. And John asked me if I would be interested in a faculty position here at UCSD, and invited me out to give a talk and visit the campus. I was then offered a tenured position here, and we accepted. Me, my wife and I. And did that for two reasons. One is, and I very much enjoyed my time at Princeton. It was great for me in many respects. But number one is both my wife and I feel very passionately about public universities and the importance and the vitalness of the public universities and the strong public universities in educating our youth. Second is that I grew very fond of living in California. While I was at Stanford, my wife's native San Franciscan, and I think those are the two major factors in coming out. Plus the reputation, even then, for young UCSD in terms of the quality of the senior people that they were able to recruit. So we moved to Bonner Hall and set out to purify this chole-1 plasmid and look at its physical properties. And at this time, Nomura Sioka, who was a great colleague of mine at Princeton, he was a product of Paul Doty's lab, and he taught whatever he knew about DNA to me, which I'm grateful for. And he had developed a column called the methylated albumin Kieselguhr column, or MAC column, that could separate DNA on the basis of GC content. We used the methylated albumin Kieselguhr column to purify a reasonable amount of chole-1 DNA. We then asked the question, is this double-stranded DNA? In which case, if we alkali denature and neutralize, we could separate the double-stranded DNA into single-stranded DNA strands. It turned out that when we did alkali denature and renature, not all of the DNA was converted to the single-stranded DNA form, which showed a density of 1.722. but a lot of the DNA retained its original density. Tom Roth was an assistant professor in the department, an electron microscopist, the same facility as now I have in Bonner Hall. Tom helped us prepare the DNA and look at it and by this Kleinschmidting technique. And indeed, the coli-1 DNA was circular. And most of it was in a supercoiled DNA form. A lot of it had opened. And there were not only monomers of the DNA, but also dimers and some trimers. Its contour length is 2.3 microns, molecular weight of 6.6 kilodaltons. And its first demonstration of the circularity of a plasmid. Fred Hickson was a postdoc in my lab, followed this up with isolating a bacterial sex factor, which is actually a combined sex factor and coulsonogenic factor, and carried out a similar experiment, separating this DNA on an equilibrium gradient, alkali denaturing, finding that this DNA, which again is at 1.710, not all of it was denatured. A lot of it retained its double-stranded character, whereas the Proteus mirabilis DNA was totally denatured. Looking at this in the microscope, this DNA is 144 kilobase pairs, characteristic of sex factor DNA, and one could determine its contour length for the open circular form, and of course, much of it was in the supercoiled DNA form. Also at this time, Jerry Vinograd at Caltech had devised this technique of lithium bromide cesium chloride gradient centrifugation, where he showed that if you put this intercalating dye, lithium bromide, into the gradient, it will intercalate within the stacked bases of DNA, and it'll reduce the buoyant density of the DNA. But supercoiled DNA cannot intercalate as much lithium bromide as linear open circular because it's constrained by its superhelical nature, and therefore it's somewhat heavier in buoyant density. So this, Michael Bazarel, who was one of 3 graduate students who joined me shortly after I arrived here, and 3 of a long list of very talented, excellent graduate students that I've had in my laboratory. Michael worked out a quick technique based upon the Jerry Vinograd lithium bromide method that could quickly lyse the bacteria, and running it on this gradient could get large amounts of pure plasmid DNA. Harvey Hirschman and Stanley Schwartz purified coulsins and showed that the coulsin that's encoded by the coulsonogenic plasmid is a simple protein of a molecular weight of around 60,000. And then at this time, Don Kuhl joined my laboratory as a postdoctoral fellow. And he developed a technique called the cleared lysate technique, which was used extensively by plasmid people for many years after. And it was based upon a technique that Bob Sinsheimer had developed working with Phi-X174 DNA to purify DNA with a protein that's tightly associated with the DNA. And when Don ran the coli-1 DNA in a sucrose gradient, it ran somewhat faster, 24S, than bare coli-1 supercoiled DNA. He then treated this DNA protein complex with sodium dodecyl sulfate and stripped the protein off of the DNA. And lo and behold, what happened is that much of that supercoiled DNA was now converted to open circular DNA by treatment with STS. And looking at it in the microscope, this is the untreated DNA. This is the coli-1 DNA associated with that protein. We call this a relaxation complex. And in summing up, a lot of work on the relaxation complex. This is a somewhat crude diagram taken from a very early review article. And given limited time, I'll just sum up our studies here. If I say our studies, I mean the work of Don Kuhl, Don Guiney, who is professor in the Department of Medicine here at UCSD, Mike Lovett, Don Blair, Janko Cooperstock Portnoy, and Dave Sherritt. The complex consists of 3 proteins, when we proposed, at least in vivo, there's a cytoplasmic signal, but in vitro, when this complex is treated with sodium indodecyl sulfate, these 2 proteins, they're 16 and 10 kilodalton in size, respectively, are dissociated from a nickase, which is inhibited from activity in the presence of all 3 proteins. The nickase nicks the chole-1 DNA at a specific site, covalently binds to the 5' end. That's what Don Guiney established, the 5' linkage of this nickase. We considered 2 possibilities for what this relaxation complex does. Is it involved in conjugal transfer of plasmids, which is a rolling circle mechanism, where this protein acts as a pilot to direct the DNA from the donor bacterium to the recipient bacterium. Or we considered the possibility that it's involved in replication, via rolling circle replication, or as a nickase and ligase, excuse me, and an intact theta replication molecule. Well, this problem was solved because Dave Sherritt went back to England, set up his own lab, and conclusively proved that the relaxation complex is involved in conjugal transfer. And we ruled out the rolling circle replication mechanism. By we, I mean Mike Lovett did, along with Leonard Katz, where they took replication intermediates of chole-1 DNA, cut them with a single cutter, ECOR1, and then examine the cut molecules by microscopy. And by arbitrarily kind of lining these intermediates up, one can see that replication starts at a fixed site, and the loop is extended until pretty much all of the molecule is duplicated. And what's important here is that one arm of these replicating intermediates always stays the same size, and that's consistent with unidirectional replication. But not all plasmids replicate by a unidirectional mechanism. Mike Lovett, Bob Sparks, and Roberta Coulter showed with plasmid R6K that it replicates by a bidirectional replication mechanism. From the origin, there's replication in the counterclockwise direction and stops at a terminus. And then it moves in the other direction to complete replication of the circle. Roberta Coulter isolated this fragment with this terminus and showed that it'll work regardless of what origin of replication we're talking about. If you take that terminus and put in chole-1, it converts it from a unidirectionally replicating molecule to a bidirectionally replicating molecule. Now, Don Kuhl went back and looked at these relaxation complexes and how they're formed. And one of the things that he did was he asked chloramphenicol, the relaxation, the bacteria, that have the relaxation complex, allows the bacteria to sit with this chloramphenicol. He shuts down protein synthesis. What happens, the formation of a relaxation complex? What happens to replication of the plasmid? And astoundingly, the plasmid chole-1, in the presence of chloramphenicol, the relaxation complex, of course, the amount that's there doesn't increase, and the protein synthesis is inhibited. But the plasmid continues to replicate until after 18 hours, starting with 10 to 15 copies of the plasmid per chromosome, one finds something like 1,000 to 3,000 copies of this plasmid in cells that have been allowed to sit in the presence of chloramphenicol. It was later shown that plasmid chole-1 does not require new protein synthesis to continue to replicate. Don Blair, who was a chemistry graduate student who joined my laboratory, looked at this chole-1 DNA that was made in the presence of chloramphenicol. He did the standard, treat with alkali, neutralize, is it still supercoiled? Well, to our surprise, when he carried out this treatment, much of that DNA denatured. Secondly, when he treated with ribonuclease, also much of that DNA was converted from supercoiled DNA form, shown here with these dashed lines, treat with ribonuclease for 10 minutes, and much of the supercoiled DNA is converted to open circular DNA, as with this graph here. The DNA, chole-1 DNA that's not made in the presence of chloramphenicol is not affected by this ribonuclease treatment. Well, much more was done with this system, and we finally came around proposing that what's in this chole-1 DNA, when you make it in the presence of chloramphenicol, are residual ribonucleotides from Okazaki fragments in replication, lagging strand replication, and from primer RNA. And so you find both strands contain it, and that in the presence of chloramphenicol, the bacteria are very bad about removing those ribonucleotides. But if you take that plasmid DNA that's made in the presence of chloramphenicol, it still has very good transforming activity. And because you put it into cells, not treated with chloramphenicol, they repair that DNA, remove the ribonucleotides, and you have then the characteristic chole-1 DNA. Okay. Now, at this time, in 1973, Stan Cohen, Chang, Herb Boyer, and Bob Helling published a classic paper where you could take plasmid PSC 101, which was made by Cohen, and you could introduce foreign DNA into that plasmid, and that foreign DNA will be maintained. We reasoned that we should try to develop the chole-1 as a cloning vector because it's present in many more copies per cell than PSC 101, and one can generate large amounts of that DNA by chloramphenicol treatment. So we joined up with Herb Boyer, who provided the EcoR1 for this experiment. By we, I mean Vicki Hirschfield, a postdoc in the laboratory, and Mike Lovett. And we collaborated with Charlie Yanofsky to show the advantages of working with the chole-1 vector. So Charlie provided us with the strains and the assay conditions. We found that normally it's an E. coli strain, W3110. This is the level of anthranilate synthetase that Stanley Mills has worked a lot on, and tryptophan synthetase. If you introduce a plasmid here, the chole-1 plasmid, which has the tripoperon inserted at that EcoR1 site of chole-1, you find that under tight regulation, you get an increase of about 40-fold of the synthetase and the A protein, about 18-fold. And then if you de-repress this system by adding indolacetic acid, you get an enormous increase in level of enzyme in the bacteria. 25% of the bacterial protein represent these tripoperon enzymes. In addition, if you allow this plasmid to replicate in the presence of chloramphenicol, 45% of the total DNA is this plasmid. So it's a very rich source of whatever DNA insert you have put into the chole-1 plasmid. Now, in the course of that work, Vicki Hirschfield noticed that this plasmid is not fully stable. And what happens is that small derivatives spontaneously arise to a recombination event, and we call this a mini chole-1. It's an agarose gel electrophoresis. That mini chole-1 is much smaller, about half the size of chole-1, runs much faster than the chole-1 plasmid. Mike Lovett generated a derivative of that mini chole-1, which is PML21. So we felt this was even a better vector. There was less vector DNA there. Shortly after that, Herb Boyer took a member of the Coley-1 family, and he treated it with EcoR1 star, which broke the Coley-1 into a number of fragments, and randomly reassembled them. And voila, he came up with the PBR plasmids, which many of you have worked with, the PBR 322, PBR 325, and so on. And then Holcomb Messing took the Coley-1 like plasmid and even reduced the plasmid to a smaller size, and we got the puck plasmids, which many of you have been working with. Mike Lovett followed this up with trying to isolate, and did isolate successfully, a replication region of an F plasmid. Taking F black plasmid, which we obtained from Francois Jacob's collection, it's 143 kb, treating with ECAR1, getting a number of fragments, selecting the replicating fragment with kanamycin, and we were able to select a fragment that's 9 kb that has the replicating functions of the whole plasmid. And we've since reduced this to even a much smaller size. At the same time, Stan Cohen and Ken Timmis at Stanford pretty much did the same kind of experiment. And I bring this up now because some 16 years later, where am I? Okay, 1976, 1992, Mel Simon very cleverly used the F plasmid to design so-called BAC vectors, or bacterial artificial chromosome, which allowed one to clone up to 300 kilobases of DNA in E. coli, fairly stable, and that had real impetus on the genomic analysis of mammalian DNA. Now, you have to keep in mind that this was all sort of new work, and it alarmed a lot of people, particularly when the Cohen Laboratory and Boyer put frog DNA into the PSC 101 plasmid and established frog DNA in bacteria. Many people said this is an unnatural event. Scientists shouldn't be allowed to do this. You're crossing species with unknown consequences. You're going to generate highly infectious organisms like an Andromeda strain. As a result, Paul Berg, in one of science's finer moments, convened at a Silomar conference where 140 scientists that had the privilege of being at that conference decided that maybe there are certain experiments that shouldn't be carried out unless we have better containment. But there are other experiments that we should be able to carry out without much containment or with not a high level of containment. And that led to an NIH recombinant DNA committee, which I was asked to be a member of, to set down guidelines. The Paul Berg guidelines had no force behind them, but NIH did. If you did not follow the guidelines for research, you ran the risk of losing your NIH or government support. So, and this is now 1977. And this, again, this cartoon, which was published in the National City newspaper. And if you can't read it back there, it says, "It's not nice to fool Mother Nature." But that sort of indicative of the concern that was still present even as late as 1977. And in that same year, Cliff Gropstein, who was chair of biology and also dean of the medical school, put together a public forum. And I was asked to debate with Bob Sinsheimer, distinguished scientist at Caltech, someone who I greatly respected. Bob felt that we should take a more slow approach, even within these guidelines, than what the guidelines allowed. And I argued on the other side. Well, to give you an indication, yet the concern in general about recombinant DNA research, this forum took place on a Saturday morning. It was a brilliant day, sunny, and 700 people showed up at this auditorium for this debate. Bob and I then went on to a couple of other academic institutions with our dog and pony show, I guess. Eventually, of course, concern was largely abated as more and more recombinant DNA experiments were carried out, and some of the products that could impact health, like human insulin, were being produced. And we switched our attention now to another plasmid system, RK2, and I'd rather quickly go over this. Why? Because it's a broad host range plasmid. It replicates stably in virtually all gram-negative bacteria. And we were particularly interested early on in Rhizobium. Rhizobium is a nitrogen-fixing bacterium. We were interested in Rhizobium meliloti, which infects alfalfa roots and carries out nitrogen fixation. We went on, by we, I mean first Richard Meyer and Dave Figursky, postdocs, showed that RK2 is an effective cloning vector. Gary Ditta joined the lab, and with Sharon Stanfield, Dave Corbin, they developed even smaller versions of RK2, which were very useful not only in Rhizobia, but also in Agrobacterium for transforming plants. And then Tom Schmidhauser, graduate student, reduced RK2 to very, very small size and showed it's able to be used as a cloning vector in a wide variety of gram-negative bacteria. This is not alfalfa and meliloti, but it's japonicum and soybean. And these are nodules for all of those who aren't farmers or working in agricultural sciences. And within these nodules, it's packed with rhizobia, and the rhizobia carry out nitrogen fixation, converting nitrogen to ammonia, and a very high energy dependent process where the plant provides ATP via photosynthesis. A key part or a requirement for the enzyme nitrogenase to work here is that the environment must be microaerobic. Well, Gary and others, Mark Better, Dave Corbin, Tyler Dillon, Sharon, and Elizabeth Wirtz, begin to study the regulation of the nitrogen fixation genes. And there are 23 nitrogen fixation or fixed genes, including HDK, which are the 3 components of the enzyme nitrogenase. Now, Rhizobium cleverly worked out a scheme where they don't want to produce these proteins unless the oxygen level is sufficiently low so that the nitrogenase would not be inactivated. And the 2 proteins, or genes too, that were identified to sense the level of oxygen and transduce this information into a response regulator protein that would turn on 2 other positive regulators to turn on all of these genes. The protein that's key here is FixL. And I thought it was an extraordinary piece of work by Marie Gillis-Gonzalez and Gary Ditta, where they purified the FixL protein and showed that it's a hemoprotein with the same visible spectrum as hemoglobin. And it's also a kinase. And this is the FixL protein with several transmembrane sites. Marie got rid of this part to facilitate purification, but retained the heme coordination. And Marie and the others, Peter Agron, Ellen Munson, Lois and Mike Weinstein, showed that in the absence of oxygen, this L protein phosphorylated itself, and then it transferred that phosphate to FixJ, which is a classic response regulator protein of a 2-component transducing system. And what was very satisfying was that Peter and Gary then showed all this in vitro. They set up a very simple system, R-meliloti RNA polymerase, a NIF-A plasmid to measure expression of the NIF-A gene, and FixJ. When they added FixL, in the presence of oxygen, there was no expression of NF-A, but in the absence of oxygen, there was abundant expression of fix-A. So it's an elegant system, very simple, no accessory proteins that explain how rhizobum eloti senses a microaerobic environment and then turns on all of these genes. During about this time, I got together with discussions with Marlene DeLuca. And Marlene, husband of Bill McElroy, who's a father of bioluminescence and luciferase. Marlene's a professor in the chemistry department, and she had a graduate student by the name of Keith Wood, who was well-versed in the biochemistry of luciferase, and I had a graduate student, Jeff DeWitt, who was well-versed in molecular genetics and cDNA formation and so on. Many of you have seen this firefly, of course. Bill McElroy was at Johns Hopkins for a number of years before going on to NSF and then on to UCSD. And he had told me that he would pay children in the Baltimore area a penny apiece for a firefly. And of course, they went out and collected large numbers of fireflies and provided for McElroy Laboratory, where they excised the lantern of the firefly and purified the enzyme, made antibiotic to the enzyme. So Jeff and Keith set out to clone the firefly luciferase. What they did here was to make a cDNA library from poly-A messenger RNA from the lantern of the firefly. And then they cloned it into a lambda expression vector, lambda GT11. They got 50,000 transformants, and 16 produced a protein which they could detect with luciferase antibody. And they then established the largest of these cDNAs in a plasmid with a lambda PR promoter and a lambda C1 repressor, a temperature-sensitive repressor. They put this in E. coli and then induced expression of luciferase, cracked open the cells, and then examined the extract, whether it would bioluminesce if they added ATP luciferin in the presence of oxygen, and indeed did. Okay. The plasmid is also put into E. coli bacteria and established as colonies, and if you add luciferin to these actively grown colonies, they also fluoresce with the yellow-green light of Photinus pyrrhus. And then we collaborated with with Steve Howell, and who had a postdoc, David Howell, who put the luciferase gene downstream of cauliflower mosaic virus promoter and established in leaf disc of tobacco, and then regenerate the entire tobacco plant. And when we watered the plant with a luciferin solution over a 5-hour period, lo and behold, it gives off this yellow-green light characteristic of luciferase. We then collaborated with Suresh Subramani, and Suresh used the SV40 promoter and established to see if luciferase would be functional in mammalian cells. And he worked with African green monkey kidney cells. And indeed, luciferase was expressed in these cells and it could be readily measured with a bioluminescent assay. And Suresh calculated that this luciferase assay was 30 to 1,000-fold more sensitive than using the CAT or the chloramphenicol acetyltransferase assay. I'm happy to say that luciferase is used extensively now in basic research. It's used extensively in diagnostic assays, and it also has been used to locate tumors in mice and the progression of tumors in mice, and is being tried now, at least I think they're gearing up for doing molecular imaging to pick up whether it will work in humans for locating the position and progression of tumors. I'd like to now finally finish up with a few words more about plasmid RK2. We got very interested in what drives the replication of this plasmid, and there are two key regions: an origin of replication and a replication initiating protein. Just to sum up, a lot of work. The origin consists of DNA boxes, interons, AT and GC. The replication protein itself, there are two forms, a 44 and a 33 kilodalton protein, both in the same translational reading frame. There's an internal start site within the gene to give the 33 kilodalton protein. Okay. This protein is largely found as homodimers, not much way of monomers, but Arisa Tukdarian and Sylvia Perry showed that what actually binds that origin is the monomer form of the protein. And she and Igor Konieczny and Alexander Bosina and others worked out the scheme where the plasmid, with its origin of replication, it will use the host protein, DNA A, to bind to those DNA A boxes. But really what controls the replication of that plasmid is the TRFA protein, or initiating protein, which binds these interons. As a result of that binding, there's an opening of the A plus T region. That provides an entry point for a helicase associated with an accessory protein, helicase loader. That helicase loader places the helicase in this open region. It's released, the helicase is activated, and the rest is history, because then the host replication proteins take over. And this scheme, the plasmid is under control, it's controlling its own destiny, because it's this rep protein that really determines whether initiation occurs or not. But how does it regulate the frequency of initiation? And in this case, it's not the concentration of the rep protein. But we developed a model that seems now to be holding up very well, called the handcuffing model. It's a very unusual way of looking at regulation. The monomer binds the interons. If there are a sufficient number of plasmids in the cell, the origins are handcuffed by the formation of this tetramer structure, and this is largely worked out by Mike McCackern, Risa Tukdarian, Barbara Lewis Cattell. As the cell increases in mass, these origins are dehandcuffed, they're relieved of the inhibition, and then replication occurs until the plasmid concentration builds up again. Now, this model has now been shown to hold up not only for RK2, but for other interon plasmids in E. coli like F and P1 and R6K and others. Finally, we became cell biologists, okay, and asked the question, are plasmids randomly distributed in the bacterial cell, or are they localized and clustered, as this cartoon indicates? And the RK2 is at 5 to 8 copies per chromosome, and in actively growing cells, you could have anywhere from 10 to 16 copies. The answer to that question was shown by Joe Poliano, Tan Ho, and Zhenping Zhong. So bacterial cell, the red is the membrane stain, FM464. The plasmid has LAC operators, so one can tag the plasmid with a GFP LAC-I repressor, and one doesn't find 5 to 8 or 10 to 16 copies of the plasmid, but it's more like 1 or 2 or 3 copies, sometimes 4 of the cell. This is a DAPI staining of the chromosome. This is a composite picture. And the the cluster of RK2 plasmid is present in the nucleoid. This is a control where the plasmid is not present and GFP-LACI is made and it's a diffuse green in the cell. We can move this plasmid to Vibrio cholera. Once again, it's clustered and localized. It's either in a mid-cell position or in the quarter and three-quarter cell position. Move it to Pseudomonas aeruginosa with the same result. And Joe and Tan and Zhenping also showed for PUC, very, very large copy number of PUC, but it is for most cells also clustered. And then more recently, Shi and Yao and Arisa Tukdarian showed that if you add chlorophenol, shut down protein synthesis or RNA synthesis, the clusters break down, and so you have now a random distribution of the foci in the bacterial cell. And Celine Verhulst now evidence that the replication initiating protein TRFA is probably a critical factor in clustering and localization. There's much yet to be discovered, not only with plasmids, but with macromolecules in general in bacteria. Many macromolecules, plasmids, have a signal that tells the plasmid where to go in the cell. In the case of plasmid RK2, it's mid-cell or quarter cell positions after duplication. In the case of plasmid CoLE1, it's a polar position. Something is contained within the plasmid that signals where that plasmid should reside as a cluster. And then, of course, we know very little about what drives the movement of the plasmid during cell growth and cell division. And this is a fundamental question in bacteria now. We now know that bacteria are compartmentalized. There are signals that place proteins at particular locations in the cell, but we don't know what those signals are for the most part, of what's driving the movement of those macromolecules. And I'm convinced that further work with plasmids is going to be critical in solving or answering some of these questions. My final slide. I've been privileged to have a number of graduate students and postdocs in my lab. And these are some of the people who I didn't mention. Of the graduate students and postdocs. Kelly Duran was my last graduate student, and Kelly is now in the faculty in the Department of Pediatrics. Celine Verhulst is my last postdoctoral fellow. I've been greatly privileged that over the years, graduate students and postdocs have shared with me the excitement of their discoveries during the years, and I thank them for that. I thank NIH and NSF. I had the same grant for 44 years. And I don't know if you noticed, but I spelled colicins, the French spelling of colicins, in that grant application, the first one. And I didn't dare to change it because I feared that if I changed one letter of that title, they would not consider it a renewal application anymore, but a new grant application. And so it's the same title. I thank Arisa Tukdarian, in my lab for the last 18 years, and a great associate, friend, and mentor of students and postdocs.